So I'm Diane Saunders and I'm a group leader at the John Innes Centre, and I run a research group which is really interested in studying plant diseases and looking at how we can use some of the cool and exciting sequencing technologies and apply those to improve disease management, both here in the UK and also in lots of countries around the world too. So we're really interested in lots of different aspects of studying plant disease, ultimately to have an impact on disease management. So we're interested in how the pathogens actually go about causing disease, and then looking at the various different mechanisms we can kind of unpick from the plant side of the interaction to understand how we can build in better resilience into our crops. So the major focus of the lab is to study emerging and re-emerging plant pathogens, but we do focus particularly on the wheat rusts, which are fungal organisms that cause diseases of wheat. So we have 3 different wheat rust fungi. The disease is named leaf rust, one's yellow rust, and one's stem rust. And we work on all 3 of these diseases of wheat, which are a huge problem for agriculture. And every single year in every location around the globe, you have at least 1, if not 2 or 3 of the flavors of these wheat rusts affecting our crops around the world. Really, the amount of wheat that's damaged every year by the wheat rust is around 15 million tons of wheat on a global scale every single year. So that equates to around 2.9 billion US dollars of losses in wheat, which is a huge amount of wheat that's lost to just these 3 fungal organisms. Of course, there are lots of other diseases that infect wheat, and just these 3 can create that kind of scale of loss. So we're taking lots of different approaches to try to shore up our crops and make sure they're more resilient to these fungal organisms, and also to better track disease. And this is through improvements in disease diagnostics and surveillance. And so as the fungus changes over time, it will create new strains. So these new strains can have new abilities to infect different wheat varieties that may have previously been resistant. And so then we can track the movement of any new strains. We can firstly detect that they're there and then track their movement around the globe, working with collaborators. And the way that we're doing that is by utilizing some of the advancements in genomics, so using some of these cool sequencing technologies, which are very new and exciting to be working with. And we're developing new genomic-based technology that can actually make best use of these sequencing platforms in order to create new methods that can track these strains around the globe, look at where they're popping up and kind of where they're going to move next. And so that helps us then to understand which particular wheat varieties in each region are potentially under threat from these wheat rust cereal pathogens. These wheat rust fungi, they're extremely complicated, and so they have a very complex life cycle, and they undergo asexual reproduction, so creating new kind of clones of themselves. So just through mutation, they will create new clones of themselves through asexual reproduction, and that's the way that they spread from one wheat plant to the next. So they can do this process, making clones extremely rapidly. So within a week or 2, you will get more and more of these spores being created that are representative of these asexual forms of the fungus. And so that allows the fungus to move very, very rapidly within a field, and that allows the fungus to be transmitted from one field to the next, from one country to the next, and even they can be transmitted between continents through these type of spores blowing on the wind around the globe. And also at the same time, this fungus life cycle is very complicated, so it doesn't just have this part of the life cycle where it's infecting wheat. It can also go down another path which takes it to sexual reproduction, and that path can actually then involve a completely unrelated plant, which is known as barberry. It's a hedgerow shrub that you might be familiar with. It includes many species in the Berberis genus that are infected by these wheat rust pathogens, and that allows the fungus to complete sexual reproduction by infecting that other plant, that alternate host of the fungus. And so sexual reproduction, of course, creates new genetic variants in a different way, shuffling these genomes and then creating new variants, new strains of the fungus. And these can potentially be more damaging because they have new characteristics. So that can mean that then They might be able to infect, for instance, other wheat varieties that were previously resistant now might become susceptible. And so they can be very, very damaging when they start to move around and they start to spread into new regions. And so we have to keep track of both parts of this story, this life cycle. We need to keep an eye both on the asexual reproduction, the part where it's infecting wheat and it's spreading very, very quickly between wheat plants, but also the longer part of the life cycle where it goes to sexual reproduction, using these alternate host plants, these Barbary plants, in order to create new genetic variants that could potentially be really damaging. So I think climate change is absolutely affecting the distribution of plant pathogens, and so pathogens are on the move due to climate change, and we're all experiencing the impact of climate change, and of course also climate instability, not just climate change. We need to think of this in the context of climate instability. So when it comes to the wheat rust fungi, this is influencing which of the 3 different wheat rust fungi we have predominantly here in the UK. And we've seen, sadly, that over the past decade, wheat stem rust, which is extremely damaging disease, that had, we thought, been eradicated from the UK and disappeared in the 1950s. Sadly, in 2013, this re-emerged. It popped up again in the UK, and this is likely due to potentially changes in the local climate, because this particular fungus is really interested in growing at higher temperatures, and so it's much happier at the kind of hot temperatures. It also needs a bit of moisture there, but it likes to grow at very warm temperatures, much warmer than leaf rust or yellow rust, the other 2 of these wheat rust fungi. And so the fact that we're seeing more stem rust can be linked to some of the changes that we're seeing in the climate. So for instance, we might be getting earlier, warmer summer seasons. And so that means that the plants that are out there in the field, they might still be green at the point when we hit the temperatures that are able to support stem rust infection. And of course, stem rust needs green plants in order to infect the plants and for it to prosper. And so once the plants have started to senesce, then they're past the point of being infected. So if we get earlier summers, so that means that the plants are actually green out there in the field when we hit the higher temperatures because the temperature increase is more rapid, then this can mean that we get more infection with things like stem rust. So we're starting to see that kind of switch. And then at the other end of the season, we also have this issue where a lot of our autumns are now getting much wetter. So we're seeing this kind of deluge of rain in the autumn, and that can really impact farmers' abilities to go out there and plant their winter wheat crops. So for instance, if you are a farmer and you're trying to get into your field and it's totally, waterlogged, what are you going to do? Well, of course, they can't get into their field if it's waterlogged. And so if we get a lot of downpours in the autumn time when they're trying to plant their winter wheat, they will have to stop, they'll have to postpone their planting. And so when the farmers are kind of faced with these challenges, one thing they can do is they can wait until the spring. They can wait for that deluge of rain to pass, and they can wait until the spring, and they can switch the type of wheat that they're growing. Instead of growing winter wheat, they can grow spring wheat, which is planted in the kind of after Christmas and into the spring. And so they can plant that much later once the rain has passed. But then that creates an issue, because then if we were to switch to growing more spring wheat matures much later, so it stays out there in the field green for longer. And so that could then facilitate the kind of impact of any warm temperature diseases like wheat stem rust. And so this kind of creates this problem, and it's a really tight balancing act on how we're going to deal with the changes in agricultural practices which are needed to account for climate change, and how that might influence pathogen distribution. And also taking into account that we already know that pathogen distribution is shifting based on climate change. So we also work on another project which is around disease diagnostics, which is called MARPL Diagnostics, which stands for Mobile and Real-Time Plant Disease Diagnostics. And so within this project, what we're trying to do is to improve accessibility to these phenomenal genomic-based approaches for disease diagnostics, because we know that a lot of the approaches that are being created, even those that we create in our own labs, are really reliant on really large immobile sequencing machines, and they're not accessible to the widest proportion of our communities. And so we wanted to make sure that all of our community can make the best use of the diagnostics techniques that we're creating in the lab. And so we made sure that the methods that we created through the development of Marple Diagnostics could be transferred onto a portable sequencing device, which is kind of smaller than your mobile phone. And so you can literally put it in your pocket and carry it around. And so that's really a huge difference compared to the very large sequencing machines, which are completely immobile, that we usually rely on in the lab to do kind of gene and genome sequencing. And so we wanted to create a system which is very simple for people to use. So we moved it over to this portable sequencing device. And then we created a whole system around it to do genotyping of particular rust strains. So using this system, we can determine exactly which strain is present in a farmer's field within 2 days of collecting a sample. And so this compares to the many, many months that it usually takes to work out which strain you have present in a sample. And also our partners in countries like where they have poor resources would usually be dependent on sending samples for analysis overseas. So using the method that we've created, it allows them to keep that ownership of those samples within the country, and also to process these samples so they know exactly which strains they have within just 2 days of collecting them. So this is a real revolution in the way that we're tackling disease diagnostics, and it really will have a massive impact for managing these plant health threats. And so I think with Marple Diagnostics, we've really opened a new opportunity to be able to widen the access to these kind of high-level genomic-based approaches to the widest proportion of our community, and so that the new kind of advances that we're making in the lab are directly accessible to them. So I think for me, I've always been interested in how things work. And I think that really has sparked my interest since my childhood. Is I like to know how things work. So taking things apart and putting things together is really fascinating to me. And I think it's, the fascination that we have throughout studying biology is how do things work? I mean, that's what drives a lot of us, is really understanding those critical processes, and also being, the excitement of being the first person to decipher a process for the very first time. I mean, this is a phenomenal opportunity and a privilege to be able to be that person. And so I think that's what really drives me as a scientist, is that discovery, and also now in my career, also the impact that we can have. I mean, it is fantastic to see the impact that you have of working in plant science. But I think throughout my career, the reason I decided to work in plant pathology was largely because of the people around me. So, the lecturers that I had at university were phenomenal, and I really felt that I had to move in that direction because they absolutely inspired me to take that direction. And so although, of course, we're taught many different subjects at university, it was, one particular lecturer who was very inspiring, that really I could see that I could both fulfill the things that I was most interested in by studying plant pathology, looking at the kind of curiosity-driven research, so looking at that kind of spark when you find something new for the very first time, but also achieving the impact that I wanted directly from the science that we could do. And so working on plant pathogens really intersects the two. So you're able to, study very fundamental concepts, but also at the same time know that you're only one step away from having a real impact in the world. And so I think that's what really drove me to work in this particular area. So I studied my undergraduate degree at BSc level at Exeter University. And during that time, I also did a summer studentship, which I think was a really fantastic experience and opportunity. It allowed me to test the water and see whether or not a career potentially working in a lab would actually work for me. And although the summer studentship was in an unrelated topic to what I study now and what my interests are now, I found through that experience that firstly, the topic wasn't entirely what I thought I would want to pursue in the future, but I did find that I loved being in the lab, and I couldn't imagine a better place to work than being in the lab. And so I think that studentship absolutely opened my eyes to the possibilities of what it's like to work in a lab physically, and kind of the excitement that people had around you, being surrounded by these exceptional PhD students, postdocs, and so on, that were really inspiring to work with every day. And so just spending a few weeks in the lab really, cemented it for me that I wanted to go into a career in research. And once I completed my BSc, I went straight to a PhD. So I didn't do a master's, I moved straight to a PhD position. And I worked for Nick Talbot at Exeter University, and he is a phenomenal supervisor as a PhD student. And he is very, very inspiring too, and definitely one of the reasons that I wanted to study plant pathology in the first place were because of the great lectures that he gave at university. And so I spent 4 years as a PhD student in Nick Talbot's lab. And then after that, during my PhD, I was working much more on kind of fundamental questions. So I was looking at development of fungi. So I was studying the rice blast fungus and trying to understand some of the developmental processes. And so this was maybe a little step further away from the impact that I wanted to achieve with my science. So it was really important, the work that we were doing, but it wasn't close enough to the plant pathogen interaction space. And so then I decided during my postdoc I would move to the Sainsbury laboratory in Norwich, where they really do study that interaction space and then get more involved in the plant pathogen interaction space. And so then I was very fortunate to get a postdoc position working for Sophie and Camoon at the Sainsbury laboratory, and I spent 4 years then as a postdoc working. This time I initially worked on potato late blight. So I moved from rice blast that I was studying during my PhD onto potato late blight during my postdoc position. And also during that time, I obtained a Leverhulme Early Career Fellowship. And during that, I started to then develop projects working on the wheat rusts on the side. So more working on the bioinformatic elements, so trying to integrate some of the sequencing technologies into studying the wheat rusts. And then that then kind of built the foundation that I then used to develop my own lab. When I then, after 4 years as a postdoc, moved on and set up my own lab working in conjunction between the Earlham Institute and the John Innes Centre. So I started a lab as an independent fellow in computational biology. And so I think this whole time, I think you could see throughout the career, I was really interested in plant pathology, also in the impact and also in fundamental science. So I think I still move between the two, between the excitement of the discoveries and also the immediate impact of the research. And I think these are the two areas that really excite me as a scientist. And I think I feel very privileged to have a position where I get to work in an area and in a job that I really just really find hugely rewarding and also a huge amount of fun every day. So it's really a privilege to have this position.